If you remember this picture from the previous lecture, the single line, the thin line within the double-stranded DNA is representing actually the tDNA, which will be developed from the double-stranded DNA, okay? And that region is between left border and right border, okay? So this is inside the left and right border, and this is outside the left and right border. So within the tDNA region, you don't see any antibiotic resistance gene here because this is a natural plasmid. But once we take this TI plasmid, we develop it as a synthetic one. Okay, we remove most of the genes because it's had got 155 genes, and we don't need them actually. So we make a shorter TI plasmid out of this big TI plasmid, which is wild type. Okay, so we replace some of the genes. We replace auxins, cytokinins, and opines with two regions. One of that contains an antibiotic here, and the rest of the region, which is actually the right border, comes very close to the left border. This is a small region, which then contains an antibiotic selection marker and the multiple cloning site. So all this region is gone when you prepare. So in the multiple cloning site, you insert your cassets of interest. Outside the left and right border, you clone another antibiotic gene here. Okay. Is it clear? Okay, anyway. So you have two antibiotic resistance genes, one inside the left and right border, one outside the left and right border. The outside one is used for the selection of agrobacterium on an antibiotic which is outside the border. When we transform this vector in agrobacterium, we need to select the transformates, and we grow those cells of agrobacterium after TI plasmid transformation on the media containing that particular antibiotic. Okay, and the internal antibiotic gene is there, which we take along with the tDNA to the plant cell, and that helps in the selection of transgenics. Are you clear about that? And then the virulence region is not required to be transferred to the plant genome. It is required by agrobacterium to infect the plant cell. Okay, so the plasmid is an agrobacterium containing the virulence region, and this virulence region will be used by the agrobacterium for infection of plant. This may not be essentially in the plant genome, but I will tell you later. You can put this gene, which is 40 kb region, okay, into the host genome as well, agrobacterium's genome as well, to increase the number of virulence genes in the agrobacterium for superinfection. Okay, and superinfection, why it is required? Agrobacterium normally infects dicot plants. It does not infect monocot plants. Okay? If you make the agrobacterium super virulent by putting the same set of virulence genes in the host genome as well, then the agrobacterium gets the capability to infect monocot plants as well. Okay? And that fragment which is inserted inside the bacterial genome is known as Kamuri fragment. It is generally known as Kamuri, K-A-M-U-R-I, Kamuri fragment. This is a virulence gene fragment introduced into the bacterial genome. So you should remember that whatever bacterial strain we use is modified. If we add a Kamuri fragment into agrobacterium, it becomes a modified agrobacterium strain with much more virulence as compared to the wild type. And that can be used for monocot transformation as well. Origin of replication is another. So this is kind of revealed to you that when we talk about TI plasmid, you must get a clear picture of that it has got the essential features of a basic vector. That is origin of replication, antibiotic resistance, multiple cloning site. Three things essential. In addition to that, if it is a plant transformation vector, it must have left and right borders, which generate the single-stranded tDNA. And you do your cloning within the left and right borders. Whatever gene you are working with, you put that between left and right borders so that that gene should be a part of tDNA that is moving to the plant cell. Is it clear?
Sir, I had a question.
Sure, please ask the question.
Sir, how does this tDNA region that we are cloning in this plasmid is incorporated into the plant's genome? Because,
yes,
sir, is that also a random process, right? That incorporation of tDNA.
I will talk about that in the coming slides about your question, okay, that how this tDNA goes to the plant genome, okay, and how it is integrated. Yes, it is integrated in the plant genome randomly. You cannot control the point of integration. It's a random process. It can launch on any of the plant chromosome at any of the position on the chromosome. Okay.
Okay, sir. Sir, the virulence region contains different genes, right?
8 to 11 genes. 8 to 11 virulence genes.
So, sir, if we are incorporating these 8 to 11 genes, do we have to give a separate promoter as well for these virulence genes to be expressed?
These virulence genes, they are on the plasmid DNA outside left and right border. The whole plasmid DNA does not go for incorporation into the plant genome. Okay, only the tDNA goes. The virulence genes remain within the plasmid, within the bacterial cell. They don't move. They work within agrobacterium. Okay, and these virulence region genes, they all have their own promoters. This is a 40 kb fragment and 8 to 11 genes. What does it mean? You can imagine, divide 40 by 11. It's about 4 kb or 3 kb fragments. The average size of a bacterial gene is 1 kb, average size of a eukaryotic gene is 2 kb. So if you are getting about 4 kb region, it means that, by dividing 40 by 11, I said there are 8 to 11. I have not used 8 for dividing 40, okay? Otherwise, you could have 5 kb region for getting a gene, a promoter, and a terminator. So all these complete cassettes for different virulent genes, they are in the vir region, which is 40 kb in size. So all of these are actually not required for infection. These 40 genes, they are present in the wild type strain. And they are 40 kb in size. The plasmid that we use for transformation is 10 kb. It means that a lot of virulent genes are also removed, which are not required actually. Okay? That gives you these different virulence genes that give you host range. If you are going to infect only, some of the important crop plants, and you find out these are the virulence genes which are essential for infecting cotton, soybean, okay, or potato or tomato. So you just keep those virulence genes, remove all the other ones, okay? So for different crops, there might be some different TI vectors with different virulence genes, but the most common that we use in the laboratory, they can easily infect the dicot plants. Okay, if we use a bacterial strain which has got the Kamuri fragment in it, then we can also infect monocot species. Can I move ahead?
Yes, sir.
Tell me that everybody is clear that what is tDNA. tDNA is a single-stranded DNA fragment generated between the left and right borders. Okay? And that's the fragment. Plasmid stays in the bacterial cell. Plasmid does not move away from bacteria. It's that single-stranded DNA fragment which is obtained from the Ti plasmid, and that single-stranded fragment carries the antibiotic resistance gene, one single strand, and the other genes of your interest. That single-stranded DNA moves from the bacterium to the plant genome. Your concept about this thing should be very much clear. The Ti plasmid produces tDNA. tDNA is a single-stranded DNA fragment which is obtained from the region between left border and right border. Okay, so this one is the fragment which is developed from the double-stranded DNA which has our genes of interest and the antibiotic resistance. So this becomes one of the strand coding all those things. The second strand will be synthesized within the plant nucleus. Okay, are you clear about that?
Yes, sir.
ये कौन है जिससे बहुत ड्राइंग का शौक है? And you don't do drawings on these slides, okay? So this is how the tDNA is developed. I said the tDNA develops between the left border and right border. For that purpose, last time I was mentioning about VirD2. VirD is one of the virulence gene region which produces VirD1, VirD2. VirD2 is the main enzyme or protein which is involved in the production of single-stranded tDNA. And how it is developed? I started elaborating that last time, that this VirD2, okay, which is written as D here, this is VirD2, okay? This produces a nick in one of the strands of the double-stranded DNA fragment between left border and right border. It produces nick in one of the strands, breaks the phosphodiester bond. The helicases, they come in, they start unwinding this double-stranded DNA into single strands. Okay? And VirD2 keeps on moving along with this, and when it reaches the left border, it makes another cut. A cut in the right border first, and then separation of that DNA fragment from the rest of the plasmid, and then cutting again when it reaches left border, generating a single-stranded DNA which is called tDNA. Okay, the plasmid regains its second strand by the polymerase activity of the cell. So the plasmid goes back to its double-stranded. This single-stranded DNA, which is generated by nicking of D2, VirD2, on both the left and right borders, it travels then from the bacterial cell to the plant cell. Okay, so the process, all the process which is involved in this, that how it moves. Now I'm talking about this. I mentioned you last time, VirE2. VirE is an other protein which starts binding to the single-stranded tDNA as soon as it is produced by VirD2. Okay? So that was the next slide. This is what I showed you last time. As the strand separates from the other strand, the VirE protein comes in and and starts binding to the single-stranded DNA. It completely covers this DNA all around. Okay, so this tDNA Vir-E complex is the one which goes to the plant cell, and I will continue its traveling from the bacterial cell into the plant cell in some slides which are coming up later. But before that, what is essential for all this to happen is the infection process. The bacteria must infect the plant cell. Okay? And I will take you to the infection process first, and then the migration of this tDNA to the nucleus of the plant cell. All right? So this is a structure that I showed you last time, that this is a complex of Vir-E protein plus tDNA, which is transferred to plant cell. The plant cell nucleus, the second strand synthesis takes place. So this single-stranded tDNA becomes double-stranded. And what is at the termini of this single-stranded tDNA molecule? Is it linear or is it circular?
Circular.
Why? Is there any ligase which is joining the single-stranded DNA at the terminal ends? There is no enzyme that can join the single-stranded ends. It should be linear, okay? Because there were cuts at the two ends, and there is no protein which is going to join the ends. But what are the sequences at the ends, at the terminal ends? There are sequences from left border and right border. So the termini of a tDNA, they contain some sequences from left border and right border. And these sequences, do you remember a cos site? Where did you hear about cos site? I gave you a lecture on cloning vectors.
I guess in cosmic.
Yes, cosmet is a derivative having a cos site, but the cos site is actually the 12 nucleotide overhangs at the 48.5 kilobase big lambda phage. The lambda phage has got, it's a linear molecule. At its terminal ends, it has got 12 nucleotide long cohesive ends, and these 12 nucleotides, they are known as cos sites. Why these cos sites are important? These cos sites, they are the hot spots for recombination. Okay, so they can recombine with the host genome very easily. So pushing all this tDNA inside the host genome. Okay, so how this tDNA, after getting double-stranded in the nucleus, gets inserted into the host genome? It is due to the presence of those left and right borders. Those are not actually the cos sites, but they are some other kind of hot spots for recombinations. Okay. How do retroviruses get into the human genome? Is it a part of the retrovirus, or the complete retrovirus gets into the human genome? It's a complete virus, because at the terminal ends of the virus, there are some sequences which are the hot spots for recombination. If you know HIV has got 250 nucleotide single-stranded DNA repeats at its both ends. And those 250 nucleotide repeats, they are involved in the integration of HIV into the human genome. Okay, so remember, these viruses, how they integrate, because they have very specific sequences which are hotspots for recombination. Whenever they go to genomic DNA, they can find some similar sequences or some relaxed sequences there, and they can just recombine with them. Recombine means they get into the genomic DNA by making a space between them. The terminal ends, they get integrated, and automatically every sequence which is inside the terminal ends, it gets into the genome. Okay, so think of how virus genomes they integrate. They use those terminal hotspots of recombinations, and this has got some nucleotide sequences from left border and right border. And the integration into the plant genome is through these border sequences of the right border and left border. Whatever will be in between those recombinational hotspots will be integrated into the gene. Okay? Keep in mind, if you have a doubt, ask me a question whenever you want. So, we've said we were talking about the virulence genes, and the figure is 8 to 11 virulence genes are there. The most important of them, which are actually responsible for infection and taking this tDNA to the host genome. There are just a few. Okay, where A, where B, where C, where D, where E, and where G. F and H. They also, so they are 1, 2, 3, 4, 5, 6, 7, and 8 genes. Okay. These 8 genes have subfamilies within the same cassettes. Where D1, where D2. So where D1 is not essential, where D2 is essential. Okay, for producing a NIC. So from the native plasmid, from the wild plasmid, the where D1 can be removed. Similarly, where E and where E2, where E3, where E2 is important, it is kept. The rest of the where E proteins or genes can be deleted from the TI plasmid, which is wild type. Okay, so reducing the size of the Vir region to accommodate only the genes which are essentially required in the infection process, in the tDNA development process, and then movement of the tDNA from bacterial cell to the plant cell nucleus. That's all. So now you know that what Vir D2 is doing. What is it? What is it doing?
It cleaves the double-stranded DNA.
Sorry?
It acts to cleave the double-stranded DNA, D2, Vir D2.
It's a nicking enzyme. It produces a nick in the left border and the right border sequences. So it helps in generating the single-stranded tDNA by producing a nick. I'm repeating it again and again. Okay? So it does not have to do anything with making a double-stranded DNA. It is involved in making the tDNA by producing the nicks in left border and right border. This is what I was showing you in this slide. Okay, so there is a nick here which is produced, and then the nick helps to separate this single strand from the double-stranded plasmid DNA with the help of helicases, which are unwinding the DNA to remove this single strand. And then the where D2 reaches here, it makes a cut here. And this tDNA, this is a tDNA now, which gets separated from the plasmid. And this is single-stranded. It's a linear, and its ends have got some sequences from left border and right border. And these are the sequences which help in the integration of this tDNA into the host genome. Okay, anyway, where D2 is important in producing the next in the generation of tDNA, single-stranded tDNA. This is very simple, the first step. And then where E binds to the tDNA. Okay. So that is the two important enzymes which take care of the tDNA production and then protection by covering it. Now I'm going to the infection process, and I also talked about this in the last lecture too. I was just recapping, but it has taken me a long time to make it clear again in your minds that what is going on. Okay. So when the plant cell is damaged, I mentioned that 60 to 70 or 80% of the plant cell is actually the vacuole. And vacuole has got a lot of compounds which are very toxic to the cell. Okay. And these compounds, they contain phenolic compounds. And the acidic and the pH of the vacuole is acidic, between 4.5 to 5. Because vacuole occupies 60 to 70% of the volume of a plant cell. When it bursts by damaging the cell, all those acidic compounds, they are released, dropping the cellular pH to acidic side. And one of the phenolic compound is acetylsyringion. The acidic pH and the presence of acetylsyringion actually stimulates, triggers the expression of virulence genes. Okay, so the bacteria now is capable of attaching to the plant cell wall, because some of the virulence genes, they are coding for certain microfibers of cellulose which adhere the bacteria to the plant cell. Okay, so the process is, after the plant gets damaged, pH drops to acidic side in the damaged area, and the acetylsyringion, which is released from the damaged cell, it triggers the virulence genes in the acidic pH. Okay, so the virulence genes start everything: the infection process, the tDNA formation process, and the transfer of tDNA from bacterial cell to the plant nucleus. So how this actually happens, this is very important and very interesting, in fact, that you can see that how the nature works. Vir A gene, I mentioned in this slide, there are Vir A, Vir B, Vir C, Vir G. So we are addressing each of these Vir genes one by one. So what Vir A does is, Vir A is a protein which is constitutively expressed. A constitutive promoter means a promoter which produces a protein all the time, whether or not there is an inducer or there is a trigger or no. It is just producing the protein. It is not stopping producing that. Okay. So the TA plasmid has got the Vir genes, and one of the gene on the virulence region is Vir A. And Vir A is regulated by a constitutive promoter. And Vir A keeps on being produced by the cell. Vir A protein does not live in the cytoplasm. It lives in the cell wall. Okay? It might be having some hydrophobic amino acids which take the protein to live inside the membrane where there is a hydrophobic situation. Okay. So this Vir A gene, a protein, this is shown in brown color here. Okay, this is the polypeptide of that. It is traversing through the membrane. Okay, and this is the amino terminus of this Vir A protein, and this is the carboxy terminus of this protein. Some of the protein goes outside the bacterial cell wall, okay? And this acts as a sensor. Sensor of what? Vir-A is a sensor protein on the bacterial cell wall produced by the Vir genes, which are present on the plasmid. Okay, this is a sensor for acetosyringion. When there is a damage to the plant cell, in the presence of acidic pH, this acetosyringion is detected by this Vir-A protein. Okay. On the detection of acetosyringion, what happens is the kinase enzymes which are present in the cell, they phosphorylate it. They phosphorylate this protein immediately. The phosphorylation means this protein becomes active. And then, after this protein becomes active, there is a cascade of other reactions which start after this phosphorylation process of the Vir-A. So what is the next after this phosphorylation of the Vir-A protein, which is there in the bacterial cell wall? It's the phosphorylation now of Vir-G. Vir-G is shown with a circular brown color. And what is these double line indicates? This double line indicates cell wall of the bacteria. What protein is associated with the cell wall? Vir-A. Vir-A gets phosphorylated and it triggers the phosphorylation of Vir-G protein. So, two sensors. One is on the cell wall, the other is in the cytoplasm of the agrobacterium. And where does this Vir-G come from? This Vir-G is coded by the Ti plasmid, which has got the Virulence genes on it. Vir-G is there. It is also constitutively expressed. It means Vir-A and Vir-G, they are always present in agrobacterium if you transform the agrobacterium with a Ti vector, the Ti plasmid. Okay, same thing. So the sensor tries to sense whether there is acetosyringion around, and it understands that there is a plant cell where the bacteria can infect, because the acetosyringion is released by the plant cell, damaged plant cell. Is everybody clear?
Yes, sir.
Yes, sir.
I hope so. So, Vir-A is on the bacterial cell wall, Vir-G is in the cytoplasm. The first sensing is done by Vir-A upon the release of acetosyringion by the breakage of the plant cells, damage to the plant cells. Okay, and then the signal is passed on to Vir-G, which is cytoplasmic. And then, अपना mic check कर ले सारे, please. Okay, so with this Vir-G triggers now the expression of all the rest of the Vir genes which are there on the Ti plasmid. Okay, after sensing acetosyringion and upon phosphorylation of Vir-G, all the other Vir genes become active to perform their role. Vir-D2 immediately comes in and it goes to the plasmid DNA, makes a tDNA from there. Vir-E joins the single-stranded DNA, covers it up, and makes it ready for transportation to the plant cell. Okay, now you know 4 of these proteins very well, don't you? Okay, and in the beginning, I also told you about Vir-C1 and Vir-C2. If you remember the overdrive sequence, overdrive sequence is present in what kind of strain? Nopalene or octopene? In the octopene strain of agrobacterium, there is an overdrive sequence. The octopene strains, they make two tDNA molecules. The overdrive sequence is not present on nopalene strains. Okay? So, if the bacterial strain is an octopene strain, it will produce C1 and C2, which actually interact with the overdrive sequence to efficiently produce the tDNA. Just they increase the efficiency of tDNA formation. Without these C1 and C2 proteins, still the D2 can cleave the left and right borders. Okay, but it will be relatively slowly as if this overdrive sequence is absent. And that's the reason I was saying that overdrive sequence can be cloned into the TI vector based on nopalene strains. Okay? So you can take this overdrive sequence to the nopalene strain as well to make that plasmid more efficient for making a tDNA molecule. So the C1 binds to the overdrive site to trigger VIRD2 to produce a NIC. That is the initial point of tDNA formation. Okay. C2 function is not known, but in the absence of C2, the tDNA is happily formed. So there's no reason to include the VIRC2 gene in the TI plasmid that you are using for transformation. That can be deleted from there. Okay? But if you want to use the overdrive sequence, VIRC1 should be there, because VIRC1 binds to the overdrive and then it binds to the VIRD2, and then VIRD2 makes a cut on the double-stranded DNA between the left and right border sequence. The first cut is on right border and the second cut is left border after the strand separates. Now the remaining three, where H, where F, where J. Okay. Where H actually codes a protein which is very close to cytochrome P450. You know the photosynthesis process? I hope in your early degree program, you might have heard about photosynthesis. Have you heard about photosynthesis?
Yes, sir.
Yes, sir.
Okay, photosystem 1, photosystem 2.
Yes, sir.
Yes, sir.
There is a cytochromes there.
Yes, sir. Yes, sir.
Cytochrome C there, okay? These cytochromes C, they are the electron transport chain carriers, okay? And they actually help the cells to get rid of oxidation processes, okay? Oxidation processes, they are lethal to the cell. They are very harmful to the cell. Okay, so this is a very beneficial enzyme playing its key role in the health of the cell. So the phenolic compounds, they are toxic when the vacuole bursts. They may be toxic to the other cells which have not burst out, and they may also be toxic to the bacterial cell. So bacteria has got an immunity by producing a cytochrome P450-like protein, which detoxifies these phenolic compounds so that bacteria can happily infect the plant cell. Is it clear? This is one of the important genes in the virulence system, which protect the bacteria, which neutralize the damaged plant cells, and then helps bacteria and plant to develop a very good coordination between them. Where F, it determines host range factor. Okay, so where F, I mentioned that if this considered TI plasmid is originally a dicot infecting plasmid. With the help of agrobacterium, it cannot directly infect, but with the help of agrobacterium, it can infect the plant cells very easily. And where F tells the agrobacterium that what are your most susceptible plants. Okay, so that is the host range. You cannot infect every plant with a single TI plasmid. What kind of VRF is there? Is that VRF capable of infecting cotton or not? If it is capable of infecting cotton, the agrobacterium will infect cotton. Otherwise, it will not. So, the dicot transformation vector, the TI plasmid, still has some specific host range for that, for every different kind of VRF proteins. Okay, so this VRF determines the host range, but it should always be there in the virulence region of the TI plasmid, so that it gives the capability to the agrobacterium to infect that plant. If it is not there, agrobacterium, besides sensing acetylcysteine, cannot infect that plant. So this is the kind of virulence factor for infection. Virj, this is the highly essential protein. It's T-strand binding protein. What is T-strand? That's Tdna. That when the Tdna is produced, Vir is the protein which is covering all that Tdna. You remember that, okay? And this protein DNA complex is taken from the bacterial cell to the plant cell with the help of this Virj, okay? All these proteins, all that DNA cannot move through the pores of the bacterial cell wall and plant cell wall. This is the Virj, which acts as a transporter protein to transport the Tdna complex with Vir from the bacterial cell into the nucleus. This is verbal. I have included a diagram showing all that, okay? So if you don't forget my words, you can look at the diagram, but it will remind you what I have been teaching you, okay? So now you know all those essential Vir genes, okay? There is just one more, and that is ATT region on the virulence genes. The ATT genes, especially the ATT-A1 and ATT-H, they are involved in making of the microfibrils of cellulose for attachment of the bacterial cell to the plant cell. Okay, I will show you shortly. That's very interesting. Okay, so these are all the acidic polysaccharides which are secreted by these ATT-A1 and ATT-H genes. And these polysaccharides, you know, cellulose is a polysaccharide of glucose. Okay, so that acidic polysaccharide helps the bacteria to attach to the plant cell through these cellulose microfibrils. Okay, forget the other genes, Shilby, those are not that important, but the important ones, they are the ones which are already discussed. In addition to that, is ATT-A1 and ATT-H, which are involved in binding of the bacterial cell to the cell wall so that the Tdna can be transported to the nucleus of the plant cell. Now you see this diagram, this depicts everything that I have been talking about. There is a wound in the plant cell. The plant cell gets damaged. What will happen? It will generate an acidic environment around it, and it will also release acetylcysteine. If agrobacterium is there, this is the agrobacterium cell, okay, having the TI plasmid in it. If there is an agrobacterium having a TI plasmid, it senses this acetylcysteine and the acidic pH, and then it gets attached. with the cell wall of plant. ATT genes that I just mentioned, ATT-A1 and ATT-H, they release polysaccharides. These polysaccharides, they join the cell walls of the bacteria with the plant cell wall. Okay, so this is a kind of covalent bonding between bacterium and the plant cell. And on the other hand, VIR-D2, VIR-E, VIR-A, VIR-B, and then VIR-G. All these proteins are produced by the VIR region on the plasmid, producing from the plasmid a single-stranded tDNA molecule. This tDNA molecule, I will show you on the next slide, is bound by VIR-E protein. And this complex is that taken by VIR-G through these cell wall regions by creating the pores between them so that this can travel inside and go into the nucleus. Once this single-stranded DNA gets into the plant cell, it moves to the nucleus through the nuclear pores, gets double-stranded. And as this tDNA has got border sequences from left border and right border, so it can easily get integrated anywhere on any chromosome, just randomly. This is the integration process. Okay, so all the mechanism is given here from step number 1, 2, 3, okay, and 4, 5, 6, 7, and then 8. That is the natural plasmid that the opine synthesis by this fragment, by the natural fragment, produces opines which go back to the bacterial cell, and bacterial cell keeps on growing. So that's a beneficial symbiosis kind of which is happening. But we are using this for making transgenic plants by removing the opine genes, cytokinin, auxin genes, and replacing them with the genes of our interest. Okay, so this is the overall picture of agrobacterium infection process